And welcome to another A-level biology video with Mr. Phillips. Today I will be looking at mutations with you to continue the DNA mutations meiosis module that we're currently doing. So let's start then, shall we? What are mutations, what are their importance, and how they work? So this basically, I love this slide because I found this randomly and it does sort of show perfectly what a mutation is. So basically at the top you've got a normal gene sequence, and you've got the bases of GAG, so guanine, adenine, thymine on the DNA. That is obviously making up the sperm or the egg, whichever one we're talking about. That then causes the embryo and you get a normal phenotype. Okay. However, if that mutates and let's say it goes this substitution mutation and that A becomes a T, that then is mutated. That changes that codon, which you should now know codes for one amino acid. That alters, in this case, the colour of the sperm, which alters the colour of the embryo, which alters the phenotype of the person being born. So you see, just all it is literally one base being changed that can then trigger that change, and that is what a mutation is. So let's have a look then. So what is a gene mutation? It is a random, unpredictable change to the nucleotide base sequence in a gene. It therefore causes a change in amino acids, and it can be caused by lots of mutagenic agents such as toxic chemicals, such as peroxide, exposure to bromine compounds, ionizing radiation, easy for me to say, such as gamma rays and X-rays, or high energy radiation such as ultraviolet radiation. So, and obviously lots of chemicals, sort of like the things we find in cigarettes that cause cancers. So let's keep going. And well, what actually happens then? So basically we said it's a change in the base sequence, therefore effectively changing the primary structure of the amino acids. And as we've done in recent videos, there's two different ways of showing you here. I'm just going to stick to the circle one. I think if you've got, say, U C C, okay, that is serine. Whereas if it's UC, so you've just changed that middle C to a U, you've gone to phenylalanine, I think it is. So that is an example of a mutation. It will change your amino acid. So let's keep going. So there are a couple of different mutation types, okay? Now, the ones I'm showing you are what I would call point mutations. Now, you guys don't need to know massively point mutations or frame shifts. You just need to be able to look and go, that's this mutation type, that's this mutation. So I'm going to teach you something called point mutations and frame shifts today, which is a little bit more detail than you need, but I think it will really help you understand why some mutations might cause a little difference and other mutations are absolutely disastrous. So if we have a look at this, this bit of DNA here, we've got this code CTA, okay? And we're going to use this to do a substitution. So the T and the A, I should have said there, so I'm just going to delete that line that I've drawn there. There we go. So the T and the A at the end of this one have been substituted to a C and a G, okay? So that then obviously causes a change there. Next one is an insertion, okay? And an insertion is when you add an extra organic base, okay? So in this case, they've gone from being, if you look along here, you've got CCT. They've added between the two C's a T base, and obviously now on the other side, which is inserted. Okay, and then you've got a deletion here. So if you look at this one, it goes GCT. Okay, well the C has gone in the middle, so it becomes G and T. So that's a deletion. Now, the effects of mutations then can be quite different. So if we've got here the normal example at the top of the board here, so we've got the normal DNA or nucleotide sequence, okay, you can have something called neutral mutations. Okay, and now a neutral mutation is where the one codon changes to another codon. So you have the organic base that changes, okay, so in this case it's gone from a U up here to a C here, but it just so happens that UCU, which was the original triplet along the top here, okay, or the original codon, codes for the same amino acid as UGC. So even though you've changed the codon, it still codes for cysteine. So therefore, it'd be a neutral mutation because it's had a neutral effect. It's had no effect whatsoever. You then might have one called a missense mutation, okay? And this is where you would change, because you're changing the codon again, but now instead of coding for cysteine as it originally did, it's now coding for tryptophan, so it's changing it. So missense mutation is when, because of the change in base, which changes the codon, you change the amino acid in the sequence. And then you have a nonsense mutation, okay? And basically this is where, and we talked about this last lesson, some of the codons don't code for amino acids. So UGA doesn't actually code for a new amino acid to be attached. It is what we call a stop codon. So therefore, at that point, protein synthesis will stop at that exact point. Regardless of what's after it, the protein will stop being made. So obviously that can have quite a big effect, because you could get halfway through making your protein, there's a mutation, there's a stop codon, and then the rest of the protein isn't made. So you've got neutral mutation, which is where it codes for the same amino acid, so it has no effect. A missense mutation, where it codes for a different amino acid. And a nonsense mutation, where it codes for a stop codon, which stops the protein being made at that point, okay? What are the effects of these? Well, let's have a look at this. So I gave you this to do, have a go at, but I'll go through it with you just to show you really. So here we've got the normal mutation, sorry, the normal at the top, TTA, which codes for leucine. That has been, in this top one, gone to TCA, so that is a substitution, okay, which changes that just to serine. Then you've got a nonsense one, which changes it to TAA, which is a stop codon. So then no more amino acids are added to the protein, they're all missing. Okay, then we've got a, there we go, so sorry, we've got TTG, which is a neutral mutation. So it's got leucine still being coded for, so there's no real difference. And then this one, instead of adding or substituting one, it goes, you've deleted one, so TTA becomes TA. And that has a big effect because that causes what we call a frame shift. So if you delete one of the organic bases, all the others move along, and that will therefore change every group of 3 afterwards, causing what we call a frame shift mutation. So frame shift mutations then are a bit of an extension. So this is, instead of just altering one codon, you can either delete one letter or add one letter, but it doesn't just affect one codon, it affects every codon after where the DNA polymerase is at that point. So if we have a look at this one, we've got the normal code here, okay, and on the normal code you've got TCC, okay, but they've inserted here a T. So what that means is that every codon is going to be different afterwards. So whereas this one was TCC, the next one was TGC, the next one was TAA, it's now going to be TCT, CTG, CTAAA. So you can see every codon after the one that's mutated is going to change as a result. So it's called a frame shift. And therefore, in this example, you've altered at least 3 or 4 amino acids following the mutation. So therefore, it's going to have a massive effect on the actual protein. The way I always sort of talk about this if I was in class with you is look at this. So it is a very simple sentence with the big red hen. So that's the correct sentence. If I did a substitution and I just swap the the I of big, okay, to the A there, I've only affected that one word. So I've only affected one amino acid. So I've changed big into bag, okay? The red hen is still identical. And that's like a point shift, a point mutation, which is caused by substitution of the organic base. If, however, I do a deletion and I get rid of that letter I altogether, so let me just get rid of some of this, delete some of this scribble for a second. So if I get rid of that letter I altogether, what happens then is I get this. So instead of the big red hen, it becomes a N. Okay, so all of these words have now been affected because the one letter has been deleted, and that means all the amino acids, all the bases become one closer, and therefore it alters every codon to follow. And the next one is if I add a letter. So if you remember that we had the word obviously big originally, okay, which if I add in front of the word big the letter C, as I have done here, okay, what that changes then is all the letters afterwards again. So that it becomes the big, gre, de, he, n, okay, which doesn't sound like anything as well. So addition and deletion cause frame shifts. That affects every amino acid after the mutation, and hence it has a bigger effect on the actual mutation. You need to be able to see something like this example here and be able to explain the different types of mutations that could occur and what possible effect that they would have. Okay, so let's look at a couple of examples, because these are likely to come up in the exam and they come up in sort of genetics modules later on when they link back to this. So the first thing is sickle cell anemia. So quite a common disease in parts in continent of Africa. Okay, and basically one base substitution occurs in codon 6. So across the whole of the gene, we know it's in codon 6, so the sixth set of 3 organic bases, and one substitution happens. And what it does is it alters the polypeptide chain in the hemoglobin molecule, and it causes a mutation, what we call the beta chain of hemoglobin. And basically it means instead of having glycine as the amino acid, the 6th amino acid in, it becomes valine, okay? And that has quite the effect on the hemoglobin molecule. So it's only one of 146 amino acids, so it's 1 in 146, but the hydrophobic, if you remember the structure of hemoglobin, it has hydrophobic amino acids in the middle, hydrophilic on the outside, maintaining that globular protein shape. Glutamic acid, sorry, I said the wrong one earlier, is hydrophilic, so that's what it should be, but it mutates because of the change in one organic base in the codon 6 to valine, so it becomes hydrophobic. So what happens if you think about it? What that's saying is instead of having on the outside a hydrophilic hydrophilic amino acid which is pulling outwards, it becomes hydrophobic, and as a result, you get these sickle cells start to develop called sickle cell anemia. And this only happens though when oxygen blood levels fall low and the valine forms bonds with each other and it starts to collapse. And it becomes useless, it can't carry as much oxygen, it can block your blood cells and your capillaries up, and it becomes what we call a sickle cell crisis. So let's keep going. And now we want to look at some mutations, just to give you an idea that not all mutations are bad, some can be neutral. So neutral means they've got no benefit or no selective advantage or disadvantage. And there's some of these you'll be more familiar with, some of them you won't. So there is a plant called the honeysuckle, which some people can't smell, some people think smells disgusting. And it is just literally that there is a chemical in there that's dangerous, but there's so much, you need to have so much of it that you can't, it wouldn't hurt you. So basically it's a silent mutation. Okay, there's certain drinks out there, certain chemicals that some people can taste, some people can't. They do think that affects even if you like vegetables, for example, and even rolling your tongue like that, okay, that is a mutation, however you can or can't do it, and it's just a silent mutation where the base is substituted for another one. Sorry, not silent mutation, it's a neutral mutation, because it doesn't have any benefits to us. There are also then these silent mutations, as we talked about, where you get one organic base replacing another one, but they happen to code for the same, the codon happens to code for the same amino acid. The environment is the final thing that really determines whether a mutation is beneficial or harmful. And this really is a good example of this, because this obviously has developed into skin colours. So skin colour, for example, obviously from the palest sort of skin colour to the sort of darkest skin colour, you can see on this graph here, it shows you the sort of difference in the variance of skin colour. And you can see on the map of the world, and this is sort of where evolutionary these skin colours have developed, where you can see, and it sort of shows you that the environment does determine whether that mutation is beneficial or harmful. So what would happen is if you've got darker skin, it literally is caused by one pigment called melanin, and that you've got darker skin, you've got more melanin. Melanin, which is a really useful chemical, it protects you from UV radiation, but it means you can absorb less sunlight. The people who have lots of melanin are better sort of adapted to high sunlight areas, because that means you can still get some vitamin D, but also it protects you against sunlight and skin cancer, things like that. So you have better survival chances with dark skin near the equator. Whereas if you've got pale skin, you've got less melanin in your body, and therefore you would have very little UV protection, and that's why really pale people tend to burn quicker. But you will absorb vitamin D even in lower levels. So let's say someone that lives in Canada or Alaska or something like that, they'll often be really pale, and that is because they will need to absorb as much vitamin D as they can, and obviously they've got very low sun levels. So the environment really decides whether your adaptation is suitable. Obviously, that's the evolution which you want to. Now, in terms of chromosome mutations, there are whole chromosome mutations that can happen, and they can happen in a couple of different ways. You can get changes in chromosome sets, which is like is it what we call polyploidy, and it might be that in some cases, it does happen in plants sometimes, you might get a pollen grain that instead of having haploid number cells as a diploid number cells, so you end up with, instead of diploid, triploid. And some plants can survive, but it would be a bit like a sperm cell with 46 chromosomes instead of 23 fertilizing a normal egg. You would have too many chromosomes. There's animals it wouldn't work, but in plant cells it can. You get a random change in maybe 1 or 2 chromosome numbers. So in humans, this obviously happens more when we have people having Down syndrome, and that is because they have this extra 1 chromosome in set pair 21 to become 3 instead of 1. And then there's changes in chromosome structure. Okay, so that can be where the actual chromosome structure alters as well, but we don't need to know that really. So this, when chromosomes don't separate properly, leading you need to have maybe 1 extra chromosome, like a Down syndrome. It is called non-disjunction, and basically it's because they fail to separate during anaphase, as you make in anaphase 1 or 2 of meiosis, and therefore you get incorrect numbers in the gametes that have made, and it's something we're not 100% certain on why it happens. And that's why everyone can have a test for Down syndrome, for example, as if you're pregnant. Now, Down syndrome, particularly pair 21, fails to separate. So one of the gametes gets an extra chromosome basically, and one of the gametes gets none. So one set of gametes will have, whoever has Down syndrome will get 21 chromosome, 23 chromosomes in pair 21. If the other egg or sperm got fertilized without that chromosome, it wouldn't survive. Okay, so that is Down syndrome, what causes that. So this is the last video in this section of the module, and I've told you at the start of the module we'll be looking at this exam question again because it entails everything. Now, it's a 6-mark question, but I think this could easily be multiplied to like a 15-mark question if they wanted. So it says, the yellow colour in peas is a result of an enzyme that breaks down chlorophyll, which is green. The Y allele codes for the production of an enzyme that breaks down chlorophyll. The recessive Y allele is a result of the mutation in the dominant version, and the recessive Y allele codes for an inactive form of the enzyme. Outline how the Y allele codes for the production of this enzyme, and explain why the Y allele codes for an enzyme with a different primary structure. So there's 2 parts to this question before we look at the mark scheme, and I want you to pause it. If you haven't, please pause it and have a go, because it's a really good question. What it says is, how does it code for the production of the enzyme? The enzyme is a protein, because all enzymes are globular proteins. So therefore, you're basically saying, how do we make the protein from the allele, from the DNA? And the next one is, why does it come up with a different primary structure? So there's 2 parts to this. This was at the 6-mark. Let's have a look at it then. So this is what I'm saying. This could be a lot bigger question. It's a really nice question if you understand it. If you don't understand it, you're going to be sort of struggling. So it says DNA code, or so this is the first part, G. Okay, so it's going to ask you about the DNA code. So the first thing you'd be expected to say is that the DNA base sequence codes amino acids. Okay, and it makes, or messenger RNA base sequences in there somewhere. Every 3 bases or every codon codes for 1 amino acid. This is a degenerative code, which means that there are more than 1 combination that codes for 1 amino acid. and you could get a substitution that codes the actual same amino acid. Then you're describing how it works. So transcription occurs, then translation. You get complementary base pairing described somewhere in there would be the mark. Messenger RNA strand is made first, and then RNA polymerase combines the RNA nucleotides together. Translation, then the messenger RNA leaves the nucleus to the ribosomes. tRNA or transfer RNA binds to the messenger RNA, codon to anticodon. Transfer RNA brings in a specific amino acid, and then the messenger RNA is therefore translated into a polypeptide. The amino acids stick together through peptide bonds. Now, why would it change from one allele to another? Because there is a substitution, and that causes either a frame shift, but I'd prefer you to just say a substitution. Okay, that causes a different sequence of DNA. That causes a different messenger RNA codon, which means a different tRNA anticodon combines, and therefore a different amino acid attaches. Okay, you therefore end up with a different sequence of amino acids, and therefore you end up with an amino acid primary sequence or a primary sequence of protein that is different, which would then cause you to code for a protein that has a different primary structure. So I hope that's been useful for you guys. Let us know if you need any help. Other than that, see you soon.